on biology. So this is the continuation from Meiosis Part 1. So Meiosis Part 1, we walked through like how meiosis happens. So meiosis 1, the splitting of the homologous pairs, and then meiosis 2, the splitting of the sister chromatids. So we walked through that process last time. So this is going to explain how meiosis creates genetic variation. So this is where we're going to talk about that crossing over part of this and this recombination of genes. So let's get into it. So again, mitosis produces daughter cells that are identical to the parent cell, right? They're genetically identical, right? We replicate the DNA. Each cell then gets an exact same copy of the DNA. Meiosis, on the other hand, results in genetic variation because of the crossing over. Okay, and I'll explain that. But what happens, so in the end, the four daughter cells, they are different. They are not genetically identical to the parent cell. So each daughter cell is different because in crossing over, right, because the tetrad forms and at the synapses, or where the two tetrads or the two homologous pairs kind of cross over on each other, these genes can swap. So genes from one homolog can swap with a gene from another homolog. So because of this, because of the crossing over and then the fusion of the sperm with the egg, the fusion of the two gametes from a zygote, we create unique offspring, genetically unique offspring. So let's talk about some of these ideas that are at play here. One is this independent assortment idea. So there's two. So during metaphase 1 with the tetrad, right? There's two equally probable arrangements that can happen when the chromosomes line up in the middle. There's two equally probable. So remember, like these are both the same chromosome. So these are both like chromosome 1, chromosome 2 that form the tetrad. So they either line up like this or they line up like this. So because of that, then when we go through metaphase 2, we run through the rest of the process. Metaphase 2 happens, then we get this, and then in the end for the gametes, we have 4 different combinations that are each as probable, right? You have a 25% chance of each of these happening. Okay? So that's this independent assortment idea. And we're going to see this again at play when we talk about Mendelian genetics and the Punnett squares. We can actually tell how many possible combinations there are. And it's really straightforward. So remember how I talked to you before about how diploid cells are 2n, with n being the number of chromosomes. Well, for the number of combinations, the formula is 2 raised to the power n, with n still being the number of chromosomes. So in this possibility, we only have 2 chromosomes, right? We have chromosome 1, chromosome 2, all right? So 2n, 2 raised to the second power, is 4. So we have 4 possible combinations. And we can apply that to human cells. So in human cells, we have 23 chromosomes, right? So our formula 2 raised to the power n, with n being the number of chromosomes, 2 raised to the 23rd power is approximately about 8 million possible combinations. So there's 8 million possible arrangements. And that's just counting just the splitting of the homologous pairs. That's counting just this process happening with each 23 chromosomes. Okay, that's not including crossing over. That can occur and change genes. That increases your possible combinations then. But just possibility of chromosomes, there's 8 million different combinations that can arise just from this. So let's talk about then crossing over. So So crossing over occurs at the chrismata, which is occurring in synapses. So let's break this down. So our tetrad, okay, this is our tetrad, our homologous pair. So the chrisma is just the site of crossing over. So this is where genes are going to cross over. And we say it's the genetic material between non-sister chromatids. So crossing over isn't going to occur between the sister chromatids on the blue or the sister chromatids on the red. Crossing over is going to occur between the blue and the red chromosome. All right, like that. So then in the end, right, let's say like these genes here for like eye color got swapped. We call those recombinant chromosomes because the chromosomes rechange and we're able to get different possibilities there. So that's what we mean by the recombinant chromosomes, right? It's just that the crossing over produces, chromosomes that have different genes that didn't normally start out with. That's all that means. Crossing over occurs. So we get a swapping of genes to occur. So let's just play with some numbers some more. So fertilization is random, right? Right? You don't control what egg gets fertilized with what sperm. You don't control that. So, and remember, each person, each cell, there's 8 million combinations. So if there's 8 million combinations from mom in the form of the egg, and there's 8 million combinations from dad in the form of the sperm, that's 64 trillion combinations for a possible diploid zygote. And that's just with 23 chromosomes. Some species have more than 23 chromosomes, which you have a lot of combinations.
Hey all, I just wanted to say thanks again for all your efforts this semester. Just what an incredible journey we have been on. I hope you are able to take some time this weekend to get some extra rest and a little bit of relaxation for yourself. So take some time to do that, clear your mind, and we will kick off on Monday with a hopeful, more smooth semester 2. Thanks, you guys.
So, yeah, 60 more trillion combinations for a diploid zygote. So that's a lot of different combinations, and that's good. That's good for our cells, that's good for the survival of our species that we have different combinations. So, the next little bit that I just want to talk about is the formation of sperm and egg because it's a little bit different. So, the formation of sperm, called spermatogenesis, follows the meiosis process exactly. Okay? So, we start with one daughter cell, okay? Our primary spermatocyte. We go through meiosis 1, and then we go through meiosis 2 to produce 4 different sperm. Okay, it follows this process exactly, and males will make sperm throughout their lifetime. So from when they're born to when they die, they will keep making sperm that entire process. For females, it's a little bit different. So the process for females is a little bit different. So for females, it's called oogenesis. So oogenesis is just the process of just making eggs. So it's a little bit different. One, so females are born with the set number of eggs that they'll have for their entire lifetime. So they don't make eggs throughout the lifetime like males do with sperm. They're born with that. And two, what happens is when females, when they make eggs, when they go through the meiosis process, they don't produce 4 haploid cells. What they produce is they produce one primary oocyte, so one primary egg cell, and 2 polar bodies. So what it is, and the reason why is because, so size comparison, sperm are very tiny compared to an egg cell. So a lot of the development and food and nutrition that the zygote needs once it gets fertilized comes from the egg cell. So that's why the egg cell itself is very big. All right. So what happens is during meiosis 1, we get a primary oocyte that occurs, okay? And this has a lot of cytoplasm. this has, most of like everything that a good zygote needs to grow and develop. And the polar body, all a polar body is just when the chromosomes split into the haploid, the chromosomes that aren't going to be a part of the main oocyte just get booted out. And then ovulation occurs, and if fertilization occurs, then we get a second polar body, which is just again just the nucleus, just the chromosomes, the extra chromosomes that are not needed get kicked out, and we get then our main zygote. So females of this process really only produce one egg cell and two oocytes. So, and the ones that get left over, we just call them polar bodies. So that's it for meiosis. So I know it's a lot, but if you think about it in comparison to mitosis, and just think about the homologous pairs just separating out, it'll be fine. So if you have questions, let me know. Other than that, I'll see you in class. Bye.